Hey guys, welcome to the first episode of VetSplained Microbiology Edition. Today, I'll be teaching you the complement system. So, what is the complement system? Let's look at the definition first. It is a complex biochemical system of heat-labile proteins. What are heat-labile proteins? They are plasma proteins that are present in your blood, and they act as a major effector for humoral and innate immune systems. So, these plasma proteins are named C1 through C9. So they are all from 1 to 9, there are different names. And it works in a cascade reaction. So one protein turns to another protein, the other turns to the third protein. So that is how it works. And all the intermediate steps have a particular jobs to do. So there are three major pathways that we'll talk about today. It is a classical pathway, alternate pathway, and the lectin pathway. So first we'll be talking about the classical pathway. So in the classical pathway starts because of antibody binding. Where does this antibody bind? So there is a protein called C1, and the C1 is divided into three different parts: C1R, C1Q, and C1S. So the antibody that is produced against a particular pathogen goes and binds to the C1Q. In turn, C1Q causes the activation of C1R, and the C1R is the one that causes the activation of C1S. So interchangeably working. And C1S is the one that first starts the pathway. So C1S causes the cleavage of C4. Now, whenever a complement protein is cleaved, it cleaves into two parts, a part A and a part B. So like you see here, 4A and 4B. And in all cases, other than one exception, the B part is always bigger, and the B part is the only one, the bigger one is the only one that goes ahead in the reaction. And the 4A, the A part, will always go aside. The exception to this is only the C2 protein. The C2 breaks down into B and A again, but here the B is the smaller part. So B does not take part in the pathway. It is only 2A that takes part in the pathway. So first talking about C4. The C4 broke down into C4A and C4B. So put the A aside, we don't have anything to do with that now. And C4B is the one that causes the cleavage of C2. Now once C2 is cleaved, what is it cleaved into? C2B and C2A. Like I told you, it's an exception. So the 2A is the one that will go ahead in the reaction. So here the 4B and 2A combine and form C4B, 2A. This C4B, 2A is also called C3 convertase. C3 convertase. Why is it called C3 convertase? Because it converts or cleaves the C3 protein. So now the C3 protein gets cleaved by the C3 convertase into what? A and B, of course. And here again, the exception is gone. So we have the C3B. So the B is the bigger one. It is the one that will go ahead in the reaction. So the C3B reacts with C4B2A, the C3 convertase, and gets added to it. So it forms C4B, 2A and 3B. So this is called the C5 convertase. Again, why is it called C5 convertase? Because it converts the C5 and cleaves it into two parts, 5A, 5B. Again, the B part is a larger part, so it will go ahead in the reaction. Now, C5B is almost the last step, after which the next steps are not really important. So the 5B reacts with C6, C7, C8 and C9, and finally it forms C5B, 6, 7, 8, 9. So this is called the MAC. MAC stands for membrane attack complex. So it is a membrane attack complex which attacks the membrane of the pathogen. So this is what causes the breakdown of pathogen, which we will see ahead. So this was your classical pathway. Let's next talk about the alternate pathway. The alternate pathway is activated by microbial cell surface. Earlier, the classical pathway was being activated by, yes, antibody. So here it is activated by microbial cell surface. There is no need for an antibody to be present. So just the entry of a microbe or a pathogen into the body causes the spontaneous breakdown of C3. Spontaneous means it just happens. So C3 is broken down into A and B. The C3b is now reacts with a factor B. So the factor B is cleaved by a factor D. Okay, no need to get confused here. A factor D, B for dog, causes the cleavage of factor B, B for ball, and breaks it down into BA and BB, like earlier, the A part and B part. Again, the B part is larger, so it reacts with the C3b and forms a C3b BB. This is also called C3 convertase here. Now, the C3 convertase here has a very short lifespan. It has a very short lifespan unless properdin binds to it. So when properdin does bind to it, it can go ahead in the reaction. If properdin does not bind to it, it will break down. It has a very short lifespan, so it won't go ahead in the reaction. So properdin or factor P has to attach to this C3 convertase, the C3BBB, for it to go ahead in the reaction. So once properdin has bound to it, it will cause the cleavage of C3 again. So C3 breaks down into A and B, where the C3b now reacts with this C3 convertase and forms C3b, BB, 3b. So this is also called as C5 convertase. Now this C5 convertase, so it will break down the C5 into again C5b and C5a. The C5b is the one that goes ahead in reactions and again reacts with C6, C7, C8, C9 and forms the MAC. MAC is the same one as earlier. So it was C5b, 6, 7, 8, 9. This is your MAC. Okay? So this is your alternate pathway. And your last pathway is the lectin pathway. So the lectin pathway has a very special beginning of the pathway. So it is activated by something called MBL or MBP. So M stands for mannose, B stands for binding, and L stands for lectin, or P stands for protein. So mannose binding lectin or mannose binding protein. So this protein has mannose getting bound to it. When mannose binds to this protein, that is the start of this pathway. So let's say the MBL and mannose have bound. Then here MBL plus mannose. This will cause a MASP enzyme 1 and 2 to come into action. So MASP stands for mannose associated serine protease. So it is an enzyme. It is a protease enzyme. So it is a mannose associated serine protease. So serine is a protein. This is what it breaks down. So these are two types. So in presence of MASP 1 and 2, MBL causes the cleavage of C4. And from here it's basically the same as the first classical pathway. It is just the first activation that differs in the lectin binding pathway. So C4 is broken down into C4A and B. Keep the A aside, we work with the 4B. 4B cleaves the C2 into A and B. Now C2 was an exception if you remember. So C2A is the one that reacts. So 4B and 2A. So it causes 4B2A. C4B2A. This is known as C3 convertase. Okay. Now C3 convertase will break down C3 into C3A, C3B. The C3B will go ahead in the reaction. So this again gets C3B added to it and become 4B, 2A, 3B. Now this is called C5 convertase. And again C5 convertase breaks down C5 into C5B and C5A. The 5B goes ahead in the reaction and with continuous binding to C6, 7, 8, 9, it causes the MAC or membrane attack complex, which is C5B, 6, 7, 8, 9. Cool? Now, we did all of these things, the three pathways. You must have learned it amazingly. But why do we do all of this? What are the functions of the complement system? Why is the complement system even there? So the first function is opsonization and phagocytosis. So opsonization is nothing, don't get confused. It's just the activation of phagocytosis. And what is phagocytosis? Phagocytosis is your immune cells eating up the pathogenic cells. So your macrophages, for example, go and eat up the pathogenic cells, any virus or bacteria. So that is what phagocytosis means. So which particular complement protein causes the phagocytosis? This is C3b and C4b. Yes, the ones that were going ahead in the pathway, the C3b and 4b, these are the ones that cause opsonization and phagocytosis. Opsonization, again, is the activation of phagocytosis. Don't get confused. Second function is cytolysis. Now, cyto means cell and lysis means breakdown. So, the cytolysis, the breakdown of cells. Which cells? The pathogenic cells, of course, not your own body cells, the pathogenic cells. So, this is done by the MAC. So, you must have been wondering what the membrane attack complex does. The membrane attack complex goes and attacks the membrane of the pathogenic cells and causes osmotic lysis. Like because of the osmotic pressure, it causes the lysis of the cells. So it also called as cytolysis. This is the work of the MAC. Your third function is B cell activation. Now what do B cells do? B cells are the ones that are responsible for antibody production. So B cells will cause antibody production and initiate humoral immunity. Your humoral immunity is the B cell mediated immunity or antibody mediated immunity. The fourth function is inflammatory response. Your inflammation is a generalized response to any irritant or any foreign object in the body. So the pathogen acts as a foreign object. So it will incite an inflammatory response. Now which complements do this? If you remember, the A complements which we had put on the side, no, they do not go into the garbage. They are here, C3A, C4A and C5A. They cause an inflammatory reaction. They are also known as anaphylotoxins because they cause an anaphylactic or allergic response. Also an inflammatory response. It's a similar thing. And the last function of the complement system is viral neutralization. And this is done by C3b and the MAC. So they neutralize viruses and break down the viruses or cause the viruses to be damaged. So that was it, guys. Hope you enjoyed and hope you learned something new. And I hope I made this easier for you to learn. See you in the next video.